Welcome to Pharmacology Outline channel. In this video, we're exploring intracellular receptors. Intracellular receptors are a class of receptors located within cells, specifically in the cytoplasm or nucleus, rather than on the cell surface. This underlying difference in location dictates their unique mechanisms of action and the types of ligands they bind. These ligands include steroid hormones, thyroid hormones, and specific vitamins such as vitamin D and retinoic acid. There are two main types of intracellular receptors. 1. Nuclear receptors. These receptors are typically found in the nucleus and directly regulate gene expression. Examples include glucocorticoid receptors, estrogen receptors, and progesterone receptors. 2. Cytoplasmic receptors. These receptors are located in the cytoplasm and translocate to the nucleus upon activation. Examples include the mineralocorticoid receptor and androgen receptor. The mechanism of action of intracellular receptors involves several key steps. 1. Ligand entry. The lipophilic ligand diffuses across the plasma membrane and enters the cell. 2. Receptor binding. The ligand binds to its specific intracellular receptor. Receptors may reside in the cytoplasm or nucleus. This binding leads to receptor activation and a conformational change in the receptor. 3. Dissociation of heat shock, proteins, and dimerization. In the absence of ligand, many intracellular receptors are bound to chaperone proteins like heat shock proteins. Ligand binding causes these heat shock proteins to dissociate, allowing the receptor to become active, then dimerization takes place. 4. Nuclear translocation. If the receptor is cytoplasmic, the ligand-receptor complex translocates to the nucleus. 5. DNA binding. The ligand-receptor complex binds to specific DNA sequences called hormone response elements, which are located in the promoter region of target genes. 6. Recruitment of co-regulators. The bound ligand-receptor complex recruits other proteins called co-activators to enhance transcription, or co-repressors to suppress transcription. These proteins modulate the transcriptional machinery. Altered gene transcription. The net result is an increase or decrease in the transcription of specific genes. 7. Protein synthesis and cellular effects. The resulting mRNA is translated into protein, leading to altered cellular function. Let's explore some specific examples and their clinical significance. Glucocorticoid receptors. These receptors are targeted by glucocorticoids, a class of drugs widely used for their anti-inflammatory and immunosuppressive properties. They are used to treat conditions such as asthma, rheumatoid arthritis, and certain cancers. Estrogen receptors. These receptors play a critical role in reproductive health and are targeted by drugs for hormone replacement therapy and the treatment of estrogen receptor-positive breast cancer. Vitamin D receptor. This receptor is involved in calcium homeostasis and bone health. Drugs targeting this receptor, such as vitamin D analogs, are used in the treatment of osteoporosis and other bone disorders. Important considerations: delayed onset and prolonged duration. Because intracellular receptors act through altering gene transcription and protein synthesis, the effects of drugs targeting these receptors typically have a slower onset, hours to days, but a longer duration of action compared to drugs targeting cell surface receptors. Complex tissue specificity. The expression of different intracellular receptors, co-regulators, and target genes varies across tissues. This leads to tissue-specific effects of drugs. Potential for side effects. Because intracellular receptors regulate many genes, drugs targeting these receptors can have a broad range of effects, leading to potential side effects. Careful drug design can help to minimize these side effects. Drug resistance. Tumors can develop resistance to drugs targeting intracellular receptors through various mechanisms, such as mutations in the receptor, altered expression of co-regulators, or activation of alternative signaling pathways. In conclusion, intracellular receptors are powerful regulators of gene expression and play a vital role in many physiological processes. Drugs targeting these receptors are important therapies for a wide range of diseases, but their complex mechanisms of action and tissue-specific effects must be carefully considered to optimize therapeutic efficacy and minimize side effects. Thank you for watching. If you enjoyed the video, please like and subscribe for more pharmacology insights. Hit the bell to stay updated. Keep learning.